Hello, everyone, and welcome to today's webinar, Evolution in the Manufacturing of Cellular Therapies, Lessons Learned, and Current Solutions. I'm Alexis Krause of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by Beckman Coulter Life Sciences. To learn more about our sponsor, please visit www.beckman.com. Now, let's get started. Before we begin, I would like to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions as you want at any time you want during the presentation. To do so, simply type them into the Ask a Question box and click Send. We'll answer as many questions as we have time for at the end of the presentation. If you have trouble seeing or hearing the presentation, click on the Support tab found at the top right of the presentation window, or report your problem by clicking on the Ask a Question box located on the far left of your screen. This presentation is educational and thus offers continuing education credits. Please click on the Continuing Education Credits tab located at the top right of the presentation window and follow the process to obtain your credits. I now present today's speaker, Neil Kyle, Global Biopharma Manager, Cell and Gene Therapy Commercialization at Beckman Coulter. Neil has a long-standing interest in the cellular manufacturing process with the overarching intent of democratization in personalized medication. He trained in bioengineering and functional medical resonance imaging at Temple University with a focus on spinal cord injury in pediatric population. His research was on non-invasively quantifying healthy neuronal tissues as a pre-op to assist neurosurgeries. At Beckman Coulter, Neil Kyle manages the global biopharma business commercializing the life sciences portfolio in the cell and gene therapy market. For a complete biography on our speaker, please visit the biography tab at the top of your screen. Neil, you may now begin your presentation.
Thank you. Thank you, everybody, for joining today. We had this opportunity to kind of talk about this new, very exciting field of cellular therapies. And with the recent success of the CAR T-cell therapy, there has been a renewed interest in bioprocessing and the manufacturing facilities required to meet these demands. Today, we want to examine some of the barriers and advantages of how to automate this manufacturing process. Additionally, we'll look into a detailed discussion of automating an autologous CAR T-cell process, a detailed discussion of looking at each step from collection to infusion, and discussing the current offerings we have as well as the lessons we have learned. So this is our agenda for today, to look at the landscape of the adoptive cellular therapy space, CAR T just being one of them, giving you guys a breakdown of the different trials that are ongoing, some of them which has achieved the RMAT status, and we'll talk about that. The different challenges we are facing today, moving into what does the manufacturing process, how is it evolving based on the lessons we have learned from the different studies and ongoing trials, moving into what does a fully automated manufacturing 2.0 look like. Then we'll dive deep into what does Beckman Coulter Life Sciences as a company bring to this application workflow, going from all the way from collection to infusion. And then we'll have some time to follow up with questions and answer some of your questions that come up. So cellular therapy is the transplantation of human cells to replace or repair damaged tissue and or cells. With new technologies, innovative products, and of course, limitless imagination, many different types of cells may be used as a part of a therapy or treatment for a variety of diseases and conditions. If you look at the slide, the potential applications of cell therapies include treating cancers, autoimmune disease, urinary problems, infectious diseases, rebuilding damaged cartilage in joints, repairing spinal cord injuries, as well as improving a weakened immune system. So this slide kind of breaks down all the different regenerative medicine companies that are working on cell and gene therapy, trying to go from the regenerative medicine advanced therapy designation or RMAT status into a fully commercialized portfolio. So this gives you a very nice breakdown of all the different companies working worldwide. So one of the interesting things that was discussed at the Alliance Regenerative Medicine meeting earlier this year in Washington, D.C., is kind of understand the different trials, the different phases these clinical trials are, and some of these companies, as I mentioned, working on the regenerative medicine advanced therapy designation with all the different cell therapy products, which might not be all the way commercialized to what we know today as Chimera and Axicell, but the companies that are working on moving things along in terms of cell, gene, and modified cell therapies. So here you see some of the already commercialized and approved therapies from FDA and EMA. Novartis' Chimera for pediatric acute lymphoblastic leukemia and adult diffuse large cell B-cell lymphoma, as well as Gilead Sciences Yescarta for adult diffuse large B-cell lymphoma. However, in December, Bloomberg reported that only 5 patients had, so December 2017 that is, Bloomberg reported that only 5 patients had received Yescarta since its approval, in particularly because there is difficulty in kind of understanding the insurance coverage for the therapy, plus the supportive care costs. One of the things that's to be noted here is as some of these big pharmas move into development of CAR T cells, the pricing concerns have also come up. So, and then you add this to the drugs, maybe in some instances, cases, potential, side effects and difficulties in manufacturing the cells. It's clear that, we have a lot more hurdles to jump before this new cell therapy can become widely available. So as we see, almost every other institution is working on a CD19 based therapy to combat leukemia, since it accounts for almost 9.4% of around 2 million people, newly diagnosed people in the U.S. just alone. That's roughly about 55,000 people. And acute lymphoblastic leukemia, or ALL, is, as we know, highly prevalent among children, as well as accounts for about 40% of the cases in adults. So manufacturing, CAR T looks great in the clinic, right? That's well understood in the labs. But how do we scale up? How do we come from the clinic and go into full-scale manufacturing setting? So at the same Alliance Regenerative Medicine board meeting in DC earlier this year, the FDA commissioner came out and said, there is a bigger need for the tool providers, companies like Beckman Coulter and others, to work hand in hand with our potential, the other companies that are working on the designated regenerative medicine cell products, to figure out the gaps, the loopholes, the challenges, and most importantly, figure out a way to decrease the cost of goods and have a more efficient supply chain to be able to bring this really wonderful living drugs to the market more easily. So this is a really well understood problem. This gives you a breakdown of the landscape, the adoptive cellular therapy, or ACT landscape. This is a rapidly evolving category of targeted cell therapies for the treatment of cancer and includes CAR T cell, TCR therapeutics, tumor infiltrating lymphocyte TILs. But at the most basic level, these adoptive cellular therapies involve genetic engineering of our immune cells, most commonly the immune T cells, outside of the body so that we can target the proteins or protein fragments on the cancer cells. Okay, moving into the manufacturing process and how it's been evolving, right? So the first thing that we need to understand is the product must be affordable for the acceptance, for public acceptance as a therapeutic option. There has been much debate on what the cost of this therapy should be, and there is a discrepancy between the expected and the actual selling price of a cell therapy. The International Society of Cell Therapy, or ISCT subcommittee, posted a survey that showed the labor and materials to be an estimated average of 61% of the total cost of goods. In terms of constraints in manufacturing that were designated as rate limiting and that contributes substantially to increased costs, the people who took the survey cited the lack of automation and manpower ahead of the other regulatory concerns or materials and even testing. So this is what a manufacturing 1.0 look like, right? So everything is very manual. We're trying to understand how we can transfer this tech from the lab setting, from an academic facility to a manufacturing setting to scale it up. At this point, it's just the very beginning of the process, and a lot of the processes are open. It's kind of complex. There's a lot of variability in the way we quantify or we want to set up a supply chain. And there are a lot of things are manual, and you need to understand how can we move from this step to maybe move into a more of a automated setup. So this is what the experts in the field think that should be the way to move forward with, right? So on one hand, you should have an integrated way of automating and closing out all the different unit operations that go into the manufacturing. At the same time, have the flexibility to change the secret sauce, or as the science evolves, to change the way you manufacture. However, make it so easily transferable so that you can bring it back into your developed or integrated operations. Because idea is to reduce the variation, increase your robustness and control. By the same time, be open to figuring out what are the new things that you are learning along the way so that you can incorporate those change in a very easy and feasible fashion. So tool providers, right? Tool providers as in people like Beck and Culture, people like myself, people like, people who are in this field working with different customers. We have this thing. We need to understand how the field is evolving, and what can we do to be able to adapt and to provide the services that we can provide to our customers in terms of making them understand how to quickly bring about transformative fixes that bring in the most amount of value so that they don't have their automated process is open enough to accept those transformative fixes, but at the same time stay closed enough to have that scale up capability. So in essence, if I always try to bring in some analogies, we need to understand where to skate to kind of beforehand determine where the puck is going to be. This is from Wayne Gretzky, and I know that really resonates in terms of what us as a tool providers we need to understand so that we can bring in the value as the FDA commissioner had mentioned earlier this year at the Alliance meeting. So this brings about some of the current challenges to cell therapy. So it can be grouped into 4 key areas: quality, scalability, sustainability, and robustness of the supply chain to manage the cost of goods. Now, while there are 2 different kind of ways that the current solutions in the market is presented, one, you have your modular bread maker model, as it's called, which kind of provides you with a better flexibility as, but at the same time, it also kind of limits you to make changes or bring about the different changes to adapt to the process as the science evolves. So although it provides a lot of yield a higher suite of productivity and flexibility, it also has some disadvantages. On the other hand, you have the integration of different smaller unit operations, which not only makes it completely automated. So you're taking the art out of the science, making it open for innovation, and that also helps you to have a kind of better understanding how to streamline your consumable usage and provide that higher scale up or higher suite of productivity. So at Beck & Coulter, we definitely believe in the integration of different unit operations, which can result in fewer touches so that you can have that increased process innovation and get that higher productivity we're looking for. And there are reasons why. We follow this principle for a lot of the different things we have learned, right? One of the things that, how does it impact the patient in terms of pre-treatment? So studies show, and there are plenty of data that show, that this one cohort of patients, of CLL patients, when they were treated with ibrutinib for more than a year, the T-cell isolations for these patients going through CTL-19 expansion, they showed much better clinical response. So, and that kind of makes it better to understand that when it comes to CAR T, that the impact of pre-treatment can have significant more value to get a better clinical response. Now, if you have a closed system, completely modular closed system, it will be pretty difficult to bring about this sort of new changes as you learn, as people learn from different trials and different studies, because those systems are modular, but definitely not open to be incorporated those changes. What else did we learn, right? So there was this recent publication where they talked about how a single remnant leukemic B cell can destroy the entire compartment, right? So how does one account for those things? So the authors concluded by saying that in terms of all the different polychromatic assays, there has to be some sort of a MRD test to get the idea for those minimal residual disease assays to look at those specific expressions to understand how can they make this better. While it might be very difficult to account for a single remnant leukemic cell, but at the same time, what the authors primarily stressed on is this process cannot be closed enough, because if the process is closed, things like this will be remiss. And we need to be aware of these and get it out into the market so that people understand that these processes need to be open-ended, at the same time closed enough to provide you the scale-up opportunities. One of the other things we learned is the quality of the starting material is very important. We need to have a better understanding of the phenotypes that are going in the raw material, the phenotype of the raw materials going into the process. And how can you assess that? There has to be a way of making sure some of these phenotypes, CD8 positive, CD45RA positive, CCR7 positive cells are available into the raw material going from after leukapheresis going into the activation stage. So the study showed that the CLL patients who had a higher proportion of these phenotypes, provided a much better clinical response when treated with the CTL-19 expansion. Now, having this on an integrated system with unit operations is easy to incorporate, while if you're working with a locked-in bread maker model, as per se, that becomes very challenging. Now, going from making a single dose to making a batch of a living drug. So this is where it's moving towards the industrialization of it. And for this reason, this is a process that needs to be well understood and a strategy for making the desired changes. And as our understanding increases, so should the ability to optimize and control the process. For example, if cells destined for a clinical trial should be consistent and of uniform population or not. Because when you go through the cell expansion process, inherently it produces heterogeneity within the batch. And most likely it is due to the cell microenvironment. So we need to figure out how we can bring about this industrialization to go into making a full-phased scale-up process and account for the different variables and factors that might be rate limiting. These are some of the key parameters that are collected during cell therapy production, from cell count, volume, and at Beckman Coulter, we are uniquely positioned to be able to offer a set of unit operations which are pretty much golden standard or in a class of its own. From viable cell count using our ViCell fully automated trypsin blue exclusion principle with the ability to optimize and analyze settings, to moving on to getting a better understanding of how can we do a label-free method understanding the change in cell volume. The multi-sizer, which is does the exact same process, it can go down to almost 0.2 micrometer in sizing range. Not only gives you size, but also gives you the delta in the change in cell volume. The aggregate detection, same thing before formulation, it's able to understand the aggregate detection. Moving into understanding the genetic expression, the surface expression of the reported gene function, the cytoflex, which is PDA technology providing exceptional sensitivity across the entire visible spectrum. And then the metabolic indicators. When you're growing your cells in the bioreactors, how do you get this in-process control completely automated? So the Metaflex provides you this bioanalyte analyzer evaluating the pH, glucose, lactates, electrolytes in a highly automated fashion. I'm going to talk about these things and how they fit into a fully automated autologous CAR T-cell process, and we'll talk about this very shortly. So to automate a process, a multi-step process like this, it's not easy, right? So it must first be broken down into key unit operations. And the granularity of these key unit operations may be limited by the bioprocess equipment available for each step of the process. But it may also reflect the current understanding of how this is worked out and likely be phased up as we learn more and more about this process. And that's why having a set of key unit operations automated onto a same platform makes a whole lot more sense. The goal ultimately is to have, as I mentioned, is the factory automation to take the idea of this unit operation to a whole new level so that we can collect all the sort of all the systems that are executing these functions and have the big data collection going on in the back end so that we can track the whole process. This is our rendition of how a CAR T-cell creation, a journey of a T-cell happens from collection to infusion, and how we provide these unit operations with all our different product lines to go after or kind of better understand these processes in detail. Because what important things to understand is, when it comes to CAR T-cell creation, the process is the product. So automation can reduce or eliminate a number of these open operations because we have to mitigate the risk of microbial contamination. For cell therapy processes in particular, the final product and cells cannot be filtered or sterilized. So microbial contamination and other product non-compliance will only contribute to increasing the cost of goods, especially when the cell product is lost. So I want to break down this particular slide and the next couple of slides to kind of walk you guys through in how we perceive this and what are the workflows we have put together to integrate each and every single one of these integrated operations into the autologous CAR T cell process. So from leukapheresis, isolation of T cells, we need to have an understanding of cell count and the viability of these living cells. There has to be a weight of rapid mycoplasma testing, utilizing qPCR, understanding the phenotype of the cell's raw materials going into the activation stage, and also, as I mentioned, account for the rapid microbial testing to ensure that we can mitigate that. This is how we do it. So I'll talk in more detail about the Biomek, but as I mentioned in my earlier slides with the ViCell for cell counting, Cytoflex for phenotyping as well as raw material testing, DURAClone, our unitized write-down reagent products, and at the same time Multisizer to understand the sizing of the cells. You're going to see these product names pop up as we move along the process, and I'll explain how they play into the field. Moving into the activation of the T cells, whether it's bead-based activators or transposons or Sleeping Beauty, a way to understand how to activate these cells based on the beads and understanding how it changes the cell volume, the delta in cell volume, and how we can characterize the different size. The Multisizer does a really good job at it, and I'm going to show you guys some data of how it's achieved. So using the Multisizer, when you're debeading or understanding the delta in the cell volume change, here we are looking at, say, at day 0 after thawing. So there exists two different populations, one population to the left consisting of primarily your debris, and the population on your right representing the inactivated cells. So following stimulation, the population on the right, which are the activated cells, they grow in volume until they reach a peak size of, say, 766 femtoliters. The population on the left, consisting of smaller particles, is not very important or relevant in terms of the decision-making process of this harvesting cells. Rather, in this case, the QC technician is monitoring when the cell volume has reached a peak, which is depicted on day 5. At day 7, the volume of cells has begun to shrink relative to what we see on day 5. And now the median cell volume is around 500 femtoliters. And by day 10, the cells have shrunk to about 390 femtoliters. So what our understanding is, and in collaboration with our partners in different farmers, and they have indicated that the cells at the maximum volume show maximum effectiveness in killing the cancer cells. Moving on from that process to the gene transfer or transfection, delivery of the viral vector, which has to be delivered in very small doses. And we have to ensure that we are swapping out the culture media in the bioreactor for enrichment of the cells. And by upscaling it by 500 microliters at a time, so that to ensure that the S phase or the growth phase is consistent. Making sure we check the health of the metabolites so that we can implement this change of the cell media. The Metaflex, as I had mentioned, plays a vital key role in this step. There are different bioprocess analytical technologies. People are even using pH strips to understand the health of the metabolites moving forward. But the Metaflex, our bioanalyzer, is able to obtain these results in as little as 30 seconds and with very little volume. Moving on to T cell expansion, we have to understand the cell volume and size analysis. And also, once we have the finished product, we have to phenotype it again to understand the cytotoxicity of these CAR T cells and how that interacts before we move into formulation. Not to mention on this step at the same time that this will need to be analyzed and the data needs to be collected and stored, whether that's to an ELIAS system or a whole gambit of data analytics architecture. Because big data is the current mantra around cell manufacturing. Because when the process is the product, any measurable change in the culture has to be recorded and corrected if needed. And then it moves into infusion. But one of the things that the big pharmas and even our partners we work with have mentioned is the manufacturing process, the manufacturing and testing processes have to be moved to clean rooms or to a controlled, non-classified spaces. And this is the only way we can work with multiple patient samples, and they can be handled simultaneously, which will then address to facilitate a high volume production. And in those regards, our MET1 clean room monitoring system plays a vital key role in its ability to be able to move things to a non-classified space. Okay, moving into how are we going to bring together all these different unit operations to the same platform. So the Biomek or liquid handler is at the heart, at the very core of all our unit operations. It has the capability to connect, to integrate multiple different automation-friendly unit operation and functional devices onto its grid so that we can offer this sort of closed enough but still open for innovation system. When we think of automated functional testing, this just gives you a representation of how you can combine from a cell washer to a cell shaker to a flow cytometer and even a liquid handler to kind of understand how we can achieve this sort of automated functional testing setup. The software capabilities of the Biomek system or liquid handling system is very versatile. It provides us with, whether you want to launch pre-programmed methods that we have developed or the protocols to run from a suite of test menus, making sure that we are able to prevent user errors and at the same time restrict methods that are available to operators and open up to audit trail methods to be verified for 21 CFR Part 11 compliance. The software capabilities like DART is sort of like at the center of the acquisition and understanding, reporting all these different data analytics that's collected. Because as I mentioned, one of the big thing for cell therapy is we need to be able to measure any change in the culture. It needs to be recorded and it needs to be corrected. A functional assay, as we know, is very laborious, it's very time consuming, it's very tedious to perform. It has so many different steps. But when we think about if we can figure out a way to automate this functional assay into simple user-friendly steps utilizing the liquid handlers with our other unit operations, it makes a lot more sense. So as we have a more deeper workflow automation understanding, we can integrate additional functions and devices. This sort of a deck space is with a grid allows for easy installation of different labware positions and accessories. So whatever the needs are, or if the needs need to change, or we have to quickly change or adjust to different workflows, whether activation or expansion, it can be done pretty easily, and the turnaround is pretty quick. The frame of these new systems provide, enhanced accessibility for more future device integration. So it's still keeping things open-ended so that we can change or adapt as the science changes or evolves along the way. So flow cytometry, if you're looking for, in a CAR-T process, looking for a specific surface marker or a reported gene expression, as well as understanding the correct cell types, isolated and enriched, flow cytometry plays a big role in the research, whether it's cell therapy research or in a GMP setting. So when we bring these two different technologies together, the configuration is able to completely automate, say, an antibody-based staining or acquisition of samples without even any user intervention. This system could pretty much run on our SAMI software to enable multiple families of plates to be processed sequentially with consistent timing, making sure all the plates are treated the same and the results can be compared across the different studies. This giving you an idea, an example of what an integrated sub would look like in terms of when we try to understand how, when we are bringing together flow cytometry with liquid handling, centrifugation, cell counting, how would it fit in the scheme, whether it's collecting up to 15 parameters or moving into acquisition of almost 49 different parameters. It has the ability to incorporate the different tools, going from understanding whether it's what the needs are. It can quickly evolve based on the instrumentation, a centrifuge, orbital shaker, a plate washer, and even incubator to ensure dark incubations when you're working with light-sensitive reagents, but still leaving deck space available and open for eventual more sample processing. So when we think of an automated functional testing, so that we can quickly characterize cell therapy manufacturing process, this is what it looks like to us, from sample prep to reagents, moving into instrumentation, as well as the big data, the metadata analysis, and doing it in a clean and non-classified room with the help of the Met1 systems. We get this sort of a fully integrated workflow, which is able to provide us with the functional tests that we are interested in doing across the board, and not just limiting it to what it has to offer today, but at the same time keeping it open enough to bring in other changes, whether that's understanding the stability or the potency, or even doing receptor occupancy or cell signaling, as well as your classical immunophenotyping assays. Looking into understanding how can we bring down the cost of goods, especially from a raw material perspective, especially from a consumable perspective, how can we streamline these? So as I said, flow cytometry plays a very, very, very interesting part, very vital key part in there. And one of the things that goes into the process is how can we quickly test the phenotype of the cells without bringing in any variability from batch to batch, as well as do a quick QC release test on the finished CAR T product before we release it out for formulation. So we have our proprietary dried down technology, which provides a very comprehensive range of immune system monitoring, and these are unitized, dried down, classified antibodies, which the steps are as simple as moving on from adding whole blood sample to these tubes or plates, adding your lysis buffer, washing it, and acquiring it on the samples. Because these tubes or plates are optimized for the identification and characterization of the cells of the immune system. So if you look at it, this actually, the way we developed these panels, it came out of the one study protocol, one study program, which is recognized as like a reference, a golden reference in multi-site standardization. It's probably the first time, for the very first time, a unified approach to characterize cellular tolerance induction. So some of these tubes, whether you want to delineate principal lymphocytes based on the subpopulation of monocytes, or understanding the late maturation stages of the B cell, to understanding the different T cells covering naive, effector memory, or even terminal differentiation stages, it provides you the flexibility to really streamline your workflow. We did a comparison between, because the idea is to reduce the cost of goods, right? We need to be able to figure out a decision on whether and when, what are the long-term consequences of whenever we are developing these therapies. So cost of goods should definitely be at the back of our head if we want to bring the overall cost down. So for that reason, we did a comparison between what does it look like between a dry to a liquid cocktail? And to get you guys a bit better understanding, this is a set of TVNK panel with normal whole blood prepared with a lysis reagent, and we kind of overlaid the two different data sets to kind of make a point that the sensitivity and the performance stays just the same, if not better. So to give you an understanding, the red overlaid on the blue on the blue. So blue would be the liquid, and the red would be the dry reagents. Hardly any difference that we can speak of. So moving into again, how do we kind of take some of these functional assays and automate it with our liquid handling platform and add in those centrifugation steps, those viable cell counting viability steps, and those flow cytometry workflow steps? So as you know, flow cytometry has a number of sample prep challenges. If you're working with an adherent cell line, the need to have single cell suspension samples means we have to have a robust trypsinization and resuspension. And most cell staining workflows require reagent exchanges and resuspension, meaning that you need a centrifugation step for these exchanges and washes. Another consideration is the limitations on sample throughput driven by whether the cytometer takes in your tubes, your samples, whether it's in tubes or plates. So here we show a 13-color panel of using our dried down reagents, and then we drop in some liquid markers, and we try to understand how is this gonna pan out when we try to do it in a high throughput automated fashion. So the idea is sort of integrating it into a liquid handling platform with multi-channel heads, or at least a span 8 for 8 different pickups, and adding in the centrifugation for the washes, incubation, as well as the shaker, and last but not the least, the cytometer, the Cytoflex, for doing the phenotypes. So why do this, right? One of the questions is, why do we need to have this sort of a fast, high throughput method? So phenotypic screening is a powerful tool for drug identification because in these assays can be run without even any prior understanding of the target and the primary cells, which closely mimic the therapeutic setting. If we have, so advanced automation and high content imaging has, well, made this possible, but these are still relatively slow. So to address this limitation, we have developed this sort of automated workflow that is dedicated to processing the complex phenotypic assays for flow cytometry. The systems can achieve a throughput of almost 50,000 wells per day, resulting in fully automated platform that enables, very robust phenotypic drug discovery. So to show you guys some data here, the sequential gating to kind of, if you look at from the blue to red, you have your CD3 positive, CD4 positive helper T cells, from blue to the CD3 positive, CD8 positive cytotoxic T cells in red. I'm not going to walk you guys through the entire thing, but it's kind of just makes it understand how we can achieve this sort of a really powerful screening technique to test whether it's testing the raw materials going into the CAR-T process or testing the finished product coming out of the process, utilizing a best of the both worlds unit operations of dried down antibody reagents integrated onto a automated platform with cell counting, viability testing, centrifugation, and as well as flow cytometry. Okay, this is kind of taking it to where we are in terms of taking this sort of the next step. So working with different collaborators. In here, they show a purified CAR-T cell exposed to the target expressing cell lines. We have 3 different constructs, long, short, and of 2 different generations. And we want to check the potency of this CAR construct activated by the antigen in situ. the CAR positive and the CAR negative, and kind of understand the amplitude in correlation with what's happening in vivo. So the couple things that mostly interest us, and I think our collaborators, is understanding the specificity, the amplitude, whether it's enhanced domain, CD28 or 41BB, and also understanding the signal intensity per cell and how it acts or against the different pro-inflammatory cytokines, whether that's TNF-alpha production, positive, negative, looking at the multi-level cytokine expression. Some sort of quick check to check the raw materials, to check the finished product, to understand the cytotoxicity of the cells before we move it into formulation. So as I started my presentation in the very beginning, and I mentioned, this is pretty clear. There are several different opportunities to work together so that we can address a plethora of different reasons, or whether that's a barrier or whether that's a advantage of how we can automate this sort of process, a very complicated process, and provide and get a better, deeper understanding of how we can maintain the consistency and tailor the automation to fit the process, because here the process is the product. So that leads us to the end of the presentation, and I guess it's time for some answers.
Thank you, Neil, for your informative presentation. We will now start the live Q&A portion of the webinar. If you have a question you'd like to ask, please do so now. Just click on the Answer Question box located on the far left of your screen, and we'll answer as many of your questions as we have time for. So let's get started. Our first question is, you mentioned about the multi-sizer technology for cell sizing and counting, but doesn't flow cytometers or fluorometric cell counters have the same functionality?
Okay, I would say great question. Yes, that's correct. Flow cytometry can provide the same functionality when it comes to cell sizing. But cell sizing isn't the only reason why multitude of this biopharma and academic labs are utilizing the multi-sizer. To kind of give you some more granularity and examples, a lot of these companies or institutions are utilizing the multi-sizer to establish some sort of a baseline, like a correlation and calibration of actual flow cytometry scattering data to actual real-world size numbers. If you take an example of any a company across different geographies, their R&D sites might have different flow cytometry technology, right? So how do we bring them to the same page? How do we ensure that they're speaking the same language? That's what a multi-sizer does. It establishes a baseline so that we can relate to it. I remember a publication from June and Gill et al. That came out last year in Molecular Biology Methods, where the lab was able to use the multi-sizer to convert flow cytometry scattering data to femtoliters, which provided the group with a metric of cells that was not influenced by different flow cytometry technologies that are available. So now they can easily move from one vendor to the other and be able to use the multi-sizer to standardize the scattering mean across different instrumentation. Additionally, the multi-sizer gives you a better understanding of adjusting the ratios of the beads to cells for stimulation purposes, and at the same time counting cells. Although flow cytometers can count cells, but many events are not necessarily cells, and hence, therefore, you need a dedicated cell counter for cell counts. Now, to Fluorometric cell counters are definitely an alternative, but the multi-sizer is preferred because not only is it giving you a size distribution, but it's also counting at the same time.
Now, Neil, our next question. How does fluorescence-based cell sorting fit into the CAR T manufacturing setting?
Okay. So I'm guessing you're referring to FACS. So flow cell sorting is routinely used in research environments to select specific cell population based on the surface markers. However, in the case of cell therapy manufacturing, flow sorting is kind of inefficient because you have to sort a large population of cells, and hence the throughput is slow. And most importantly, most of these flow cell sorters are droplet-based, so they will be susceptible to contamination. There are some closed cartridge-based systems, microfluidic-based systems that have been introduced to kind of resolve this problem, but the sorting rates still remain relatively very slow at about 30,000 events per second. The other thing, if I'm on the bench, when you're isolating T cells, we have to culture them. And sometimes it can backfire if you don't have the right amount of CD4s and CD8s. It can skew your cytokine in the cocktail. Some of the really experienced labs are not even adding any cytokines in their culture to eliminate the addition of any stimulation other than the beads. And then when you take this sort of sorting setup and take it for experimental reasons, it could also lead to some regulatory bottlenecks as well. So for that reason, flow cell sorting, although it's used in the R&D side of testing for isolation purposes, not so much for GMP. And that's kind of reason why some of these magnetic activated cell sorting setups have become popular. But even those have some disadvantages because you have to remove the beads, and that can add to overall cost. My 2 cents, in my opinion, it would be that we need to have an increased understanding of the target cells so that we can reduce the number of cells required for this treatment. But I don't think we're there yet, and maybe we'll get there and maybe even have some sort of a better technique to kind of address this gap in the industry.
And it looks like we have time for one more question, so we'll close out with this one. What, in your opinion, needs to change for hospitals and clinics to become successful decentralized manufacturing centers?
Thank you for the question. So I actually come from a family of doctors, and I can tell you this, clinicians, they want to have control over how they treat their patient. They want to have absolute control. And since now we are talking about living drugs, they also want to have control over the manufacturing of these living cells so that they can treat their patients in a timely fashion and have more of an accountability on the whole process. So for this particular reason, the hospitals want to become primary sites so that the ex vivo therapies can be performed at the patient bedside. If you think about, let's say, induced pluripotent stem cell, iPSC-driven allogeneic therapies, there are two main bottlenecks that are standing in the way of this becoming more of a pronounced, setup. First, if I in my opinion, would be cytogenetic instability. How do we manage a cohort of patients to better understand as to why, when the cure rate increases, what are the new concerns about the late effects of a genotoxic treatment? And how can we weigh against this toxicity and even long-term sequelae, as in the pre-diagnosed condition? So there needs to be a flexibility to integrate or bring in other alternative novel therapies to go side by side or in conjugation with the therapy that the clinicians are approaching. The other thing, in my opinion, which makes it difficult or is definitely a gap in the industry when it comes to hospitals, and this could be something maybe looking into the future, is postnatal banking. If we can store healthy stem cells at birth so that it can be reintroduced back into the patient with the gene editing or cell therapeutic techniques, that would be hugely beneficial, and we can have a much better clinical response when we look forward to this sort of allogeneic treatments moving forward.
Thank you again, Neil, for your time today and your important research. Do you have any final comments for our audience?
Yes. So, one of the things that really resonates with me in terms of where we are today and where we are moving forward as we go into the next generation of cell therapy manufacturing. The lab of the future will certainly reflect the current trends in bioprocess manufacturing. And eventually the path from the bench to clinic will require automation if we are to meet the GMP process criteria, but most importantly, to manage costs. Whether the automation of this process will proceed in a stepwise fashion, or will it be a stepwise fashion of like unit operations, as I talked about, or will it be completely new, novel ways of approaching this? Time will tell. But I think we are living in a time where it's definitely exciting, and it's a wonderful time to understand how else we can figure out more new and improved cell therapies. And us as tool providers, we will definitely be working with our customers and clients to meet the demands of this industry to deliver safe, effective, and most importantly, affordable treatments. Thank you.
Thank you again, Neil. We would also like to thank LabRoots and our sponsor, Beckman Coulter Life Sciences, for underwriting today's educational webcast. Before we go, I'd like to thank the audience for joining us today and for their interesting questions. Questions we did not have time for today and those submitted during the on-demand period will be addressed by the speaker via the contact information that you provided at the time of registration. This webcast can be viewed on demand through June of 2019. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, goodbye.